Just kind of waiting for everybody to show up. It looks like there's 2 missing. All right. So today I'm probably just I'll probably mess around with Mega more than anything else. Really just using it to, again, measure qualities about sequences, measuring properties about sequences. So I sent out a file that has like 10 or 11 sequences in it as a FASTA file that I'm going to play around with today, and then y'all can kind of play around with it on your own. But I'm basically going to analyze these sequences start to finish with a few caveats about what Mega is doing behind the scenes. Although, like, Mega is like kind of a great tool for phylogenetic analyses, especially for quickly exploring data. And certainly people publish studies using Mega, right? It's a no-joke piece of software. Although sometimes it's faster and more powerful to do things on a server with specifically designed tools for a certain problem. Like Mega is definitely like a jack of all trades for molecular analyses. But if we want specifics, we might use a specific software that we would find on a Linux server. All right, so I'm going to open up that file. I'm going to go track that file down, wherever it is. And yeah, PV test. And so these are raw sequence, so I can't analyze it until they're aligned. So I'm going to have to align it, bring up the aligner view. And so the great thing about like grabbing the coding sequences, right, as opposed to getting the cDNA, is that the coding sequences are already in frame. I can't tell you how much of a pain it is to find the reading frame in a sequence, right? Because remember, any DNA sequence can be read into codon 6 different ways, depending on which base we start counting from. It gets to be a pain. So if all of your sequences are out of frame, it's just going to be a terrible alignment. But if we grab the CDSs, everything's in frame, and we don't even have to worry about it. So that's one big reason why I always grab the CDSs, and we should always try to grab the CDSs. All right, so the CDSs of a specific gene, PWL1, from a diverse group of vertebrates, from sharks up to humans, right? And almost everything in between. But not exactly everything. This is just kind of a sample of vertebrate diversity. And a lot of these sequences, they come from model animals, right? Human is definitely model. A zebrafish is like the model fish. Green anole has been the model for lizards. Zebra finch is definitely the model for birds. Everybody has studied it's a very commonly studied animal, particularly the brain, right? The zebra finch brain is studied because we really care about vocalization, and zebra finches they're vocal. So we've looked at brain and what's going on in the brain to figure out how these birds more or less speak. Anyway, so I need to align these sequences, and just so hopefully this will be nice, I'm just going to do a quick alignment of DNA, and then I'm going to translate it, hopefully to demonstrate that I won't get a codon alignment. So this is not how I want to do this. This is just an example of a DNA alignment that should insert 1 or 2 gaps at a time. This is going to take a couple of seconds to do. Right, and the more sequences we add, it's always going to slow down. Like the analytics, the processes itself will always slow down the more data that we have. So I picked 10 sequences to play around with. So the amount of time we wait on programs to finish reduces. But even this, man, this is kind of slow. Let's see the consequences of this. Oh, I'm gonna stop this. I'm not gonna waste my time with this. Nope. Nope. Stop. Why? Why are you complaining at me? Stupid. 2 seconds. All right, I'm gonna skip that and just do a quick codon alignment as I should anyway. And look at that, it's done. All right, nice pretty codon alignment. There will be a couple of gaps in place. So this particular gene, like the 3' end of this sequence, is largely conserved, right? There are very few amino acid changes at the tail of this sequence because so this is part of a very conserved domain of the protein. You'll see, or I have definitely seen, like proteins, different regions of proteins will evolve differently. So like the back end, the 3' end of this is going to be pretty conserved, but the 5' end is going to be a little less conserved, right? So we see gaps, we see things missing. The 5' end, there's fewer important stuff in the 5' end of this protein than the 3' end. Anyways, all right. So if I want to do analyses on this, I'm going to go to phylogenetic analysis, go to File, Phylogenetic Analysis. Yes, that is a protein coding sequence. And it's going to open up a analytical tool that I can mess around with. All right, and so at this point, what I most commonly use Mega for is to build phylogenies, right? I want to understand how all of these sequences are related to each other. And so Mega lets us do that pretty quickly. Particularly under the phylogeny button, this is by far the most common buttons I touch. Within Mega, it's alignment, phylogeny, alignment, phylogeny. Only two things I really do with Mega. Phylogeny, and we have a couple of different options in here. Make note that the neighbor joining tree is the fastest, by far the fastest way to construct a tree. It's the simplest. It's among the oldest algorithms to reconstruct relationships. But whenever I'm playing around with data, I just want to get a quick and dirty tree. The neighbor joining is the way to go. As I mentioned, this is kind of a bioinformatics class. It's really not a phylogenetics class. Like phylogenetics is itself, a field you can get a PhD in, that alone. And so I can't spend a lot of time going over how and explaining how each method to build a tree is different from the others. That alone takes a lot of time. I'll do my best when I can, but I focus more on the tool use, not necessarily more on what's going on behind the scenes. But nonetheless, neighbor joining is the fastest, and so if we just want a quick tree, that's the way to go about this. So I'm just going to do a quick neighbor joining. And again, phylogenetics gets a little out of control quickly because we have to define a couple of parameters to reconstruct our history. And so I'm going to run through this real fast. Like I'll tell you what parameters we're going to choose. Like this is a nucleotide alignment, right? We don't want synonymous or amino acid. The model and method, these I'm going to just tell you for now to just use a chimera 2 parameter. And I might spend a second or two, if I'm allowed to draw, you two letting me draw, I'll try to explain that one out. What these different models are and what they mean. But for now, I know chimera 2 works pretty well. It's basically setting a substitution rate. It's basically saying I want to recognize that my transitions happen more often than my transversions, so take note of that when you see sequence changes. I'm keeping my rates the same, even though that's not usually true with a protein. Like evolutionary rates change among sites in a protein or a protein coding sequence. And I'm going to leave it at that. Just click go, and it should take no time at all. Look, there, it's done. And so this is kind of like a raw output. And this, as is a unrooted tree. Now I can look at this tree in different ways using my buttons above. So right now I'm looking at this as a traditional rectangular view of this tree. However, this isn't exactly how the tree looks. Let's see if I can get that blown up. That's closer to how this tree actually looks. It's unrooted. This is what an unrooted phylogenetic tree will look like. But it's still depicting what sequences are most similar to each other, right? So for example, my spotted gar and my zebra fish, right? They're going to share a common node with each other before they share a node with anything else. All of my mammals are going to be slightly clustered together, right? So I have platypus, opossum, and human. So opossum and human are more closely related to each other, and so they'll coalesce in the tree with each other faster than the other sequences. My birds do the same, green and old, my bird do the same thing. So birds are more closely related to reptiles. Yeah. But this isn't exactly an easy way to display the tree, clearly. So I'm going to go back to my traditional rectangle. And even still, like, this doesn't accurately, it mostly accurately reflects relationships, but this is an unrooted tree. So if I want to root my tree, if I want to say, okay, this is the ancestor, and this is what I'm comparing all the other sequences to, then I'm going to select a branch to represent my root. And the oldest lineage in my tree is the elephant shark. And so the side panel here allows me to manipulate the tree a little bit. Particularly, like this one right below this arrow is a reroot button. And this reroot button lets me say this is the ancestral branch, compare all of my sequences to the ancestor. And so now we get a nice tree that mostly reflects species relationships. But this is also a gene. It's a gene tree. And PWL1 is not really like a tree we would want to use to figure out species relationships. I'm cheating a little bit, but I'm also getting to the point that I want to make. Right? This data lets me do what I want with it, whether or not it's accurate or whether or not I'm using the best data for what I'm doing. But nonetheless. So this is how we have largely figured out what's related to what, is using DNA sequences. Because morphologies have issues with convergence, So two animals that may look similar to each other may not all be all that related because they've just converged on a phenotype. But gene sequences usually don't lie. And a lot of them are neutrally evolving. And so if we see more similarities among a DNA sequence, we're going to, it suggests that those two organisms are more closely related. This reflects a vertebrate phylogeny, whether we're familiar with it or not. This reflects a vertebrate phylogeny where we're saying sharks are the ancestral group. And from that point, right, we have the fish kind of as a primitive group, and spotted gar and zebrafish cluster together. They share more common recent ancestor. The clawed frog is an amphibian. Zebrafinch and the green anole share a node with each other before they share a node with anything else. The mammals share a node with each other, particularly like the monotreme is the outgroup, and human and possum. We have a placental and a marsupial. They're going to be more closely related. And the reptiles and the birds, the reptile group and the mammal group, they coalesce back to a point to a common ancestor before anything else, right? This just reflects vertebrate evolution. Okay, so if I want to save this tree, how I'm going to save it is by exporting a NUIC. So I went to File, Export Current Tree as a NUIC, and a NUIC is a text representation of the same thing. So whenever I save this tree, I'm going to select branch links. I don't care about the gene tree. I don't think I can do anything else with this. Just make sure my branch links are saved. Click OK, and then, right, so here's my actual NUIC. This is what a NUIC looks like. And I may try to draw this out in a second. But nonetheless, I actually do want to save that as a text document. Hopefully it will just let me. And so whenever I save a NUIC, just like I always depict, like if it's a FASTA file, I save it as a FAS. If it's a NUIC, I save it as an NWK for NUIC. So trees are always going to be saved as a NUIC. And hopefully that will be true whenever I actually look at the text document. Yes. Yeah. Okay. A lot of stuff. A lot of stuff that just happened. All right. We're going to see what happens. We're going to see, I'm going to pull up PowerPoint. I'm going to pull up a slide, see if I can draw on it, and we'll see what happens. All right. There's my PowerPoint slide. All right, so let's see how well this works. Models. So we have this kind of assumption, or we can recognize that any base can change into the other. So at any particular site in a sequence alignment, we can see all four bases. We can see up to all four bases present. So let's see if I can look at that alignment real quick. Come on, where are we at? So for example, here's a nice site. So here we go. See, so in this particular site, we can see a GCTCAGA. So at this particular site, we can see all four bases present. But we also may want to ask, like, what would have been an ancestral state between two sequences? So if we see a G and an A, what was the ancestral sequence? Was it a G or was it an A? And so we have these models of substitution. So how are these bases being replaced? So so we can have a C, G, TA. And recognize, so the most primitive model of substitution, when we're trying to recreate ancestral sequences or recreating what was the most likely mutation to get to this point, the simplest of that is assuming that all of these bases can change into an other at an equal rate or an equal probability. And so this model was developed in like 1969. It's called the Jukes-Cantor model, or JC69 for its date. And it assumes that these rates of mutation are all the same. So C has an equal chance of mutating into a G, as an equal chance of mutating into a T, and an equal chance of mutating into an A. And so the likelihood or the probability of one base being substituted for another are equal. So this rate of A is equal to T is equal to C is equal to G, right? They all have this equal probability of mutating into another. And so we just apply one rate of evolution for all sites, and we just would call this alpha. Right? This is a reasonably simple model. This is the simplest model because it really doesn't make it doesn't recognize biological complexities in DNA evolution. So I would definitely say the next complex model, or the next step up, is the Chimera 2 parameter. I'm still slagging on the drawing, so I may give this up. Yeah, I may give this up. Sorry, I tried. You know, I try to science everything, like change something, see what happens. But I think drawing is just out of the question, unfortunately, for this platform. Nonetheless, a Chimera 2 parameter recognizes two rates, a transition rate and a transversion rate. Because, right, remember we have our purines that are guanine and adenine, and our pyrimidines that are cytosine and thymine. And so we've already seen before that transition already happened way more commonly than transversions. So a Chimera 2 parameter recognizes that. So if it sees a site in the alignment, if we see a site in alignment, right, we see this G and an A, it recognizes that this transition is more common. So I don't know. It's hard to explain stuff without drawing, and I really can't draw, so this is kind of this isn't going very well. Nonetheless, what we can do. Ultimately, the model of substitution matters, because if we change our substitution model, then the tree may change, because each model assumes different rates of what one base can change into another. You can get rid of that Newick. I'll leave that tree there. Cancel. All right. but I can at least draw out one more tree, or at least go through another algorithm to make trees. Again, in phylogeny, so I've already said that the neighbor joining is the simplest method. A more complex method is a maximum likelihood tree. And so maximum likelihood, there's a big difference between a neighbor joining and a maximum likelihood. So a neighbor joining is just going to recreate a tree given the sequences. Right? It's going to take that sequence information and just try to develop a tree based off of what's present. But a maximum likelihood approach addresses this a little bit differently, because a maximum likelihood doesn't try to rebuild a tree given the data. It's going to ask, what is the best tree given my data? So not what tree can I build from it, but what is the best given this data? And so maximum likelihood, it's a little bit slower. It's a little bit more sophisticated in how it finds the best tree. But nonetheless, if we're just running this piece of software and we want to build a maximum likelihood tree, I'm going to keep the model a little bit simple, a little simpler than it actually reflects. But nonetheless, my model substitution, I'm going to move to a general time reversible. And so it's also called a GTR. And a GTR is effectively an overall model, as opposed to like applying one rate of evolution for all bases, or just two rates for a transition and a transversion rate. It applies a separate rate, or it estimates a separate rate for A to T, for A to C, for A to G, right? We get three different rates out of that. But the reversible part is that T to A will be the same rate as A to T. A to C will be the same rate as C to A. And A to G will be the same rate as G to A. So we have this, we can apply different rates of evolution or different rates of substitution for every possible change, but it's the same rate forwards and backwards. Anyways, this is kind of like an overarching model, and any of the other models are effectively sub-models of it. It's just the same model with a different parameter, right? A K2P is a GTR, but G to A is one rate, A to G is one rate, and it just so happens that C to T is going to be the same rate as A to G, right? It's still the same thing. Nonetheless, it's kind of, again, it's just kind of this overarching model. And if there's kind of a rule of thumb, kind of a shortcut, I should say, when in doubt, just pick GTR and be done with it, right? It's going to work. Don't worry about it. Just pick GTR. There's dozens of papers that have said just pick GTR. And even when we try to compare these models to say what's the best one, GTR comes out pretty often. So GTR, I'm going to make a simple assumption that all of my substitutions are evolving at the same rate. Not true, but nonetheless, I'm going to make that assumption. And then click the Go button. And so this is going to be a little bit slower, not much slower, but a little bit slower. And I should get the same tree. So we've basically applied two different methods to build a tree. And if we effectively get the same method, we build the same tree using two different methods, then we're pretty confident this is an accurate tree. But even recognize we can't actually go back in time and figure out what's related to what, right? We can't go back in time and actually observe diversification events or diverging events. And so no matter what, the best trees with the best software are still kind of hypotheses. They are in themselves supported, but they are nonetheless hypotheses because, again, we can't physically go back in time and verify these results. But nonetheless, it is nice. It's very, very convenient, and it's very friendly when you can get two different pieces, two different techniques, and build the same tree, right? Because this is the same tree. Gar and zebrafinch are clustered together. Clawed frog is kind of the outgroup to all the other amniotes. Zebrafinch and green anole are sister. Human and opossum are sister, right? It's the same tree. That's nice. Let's see. Now, I have not actually. One thing I have not done is actually test the robustness of this tree. I haven't applied any kind of statistics to it. Yeah, I've not applied any statistics here. So I can at least, the only thing I can really do statistics wide in a tree is do a bootstrap. And so bootstrap is kind of like resampling the data with replacement. And the data are going to be columns. So I'm going to close this real quick. I'm going to get rid of that. I'm going to get rid of this. Okay, and so whenever we bootstrap, right, this is our data. And a bootstrap is it's really not that common of a statistics test. But if we have low sample size, we may bootstrap our data to build support in it. And so in bootstrapping in phylogenetics, we have this whole alignment, right? This alignment is 2,900 bases long, or 2,900 positions long. Now we're going to get rid of gaps. Phylogenetics, we really can't model gaps very well. We can model substitution changes, but we really can't model gaps. So most gap sites are already eliminated before we do phylogenetics. If they're not, they're typically removed. But nonetheless, whenever we bootstrap, we're going to take this raw data and we're going to repeatedly sample from this data to make a new alignment. So for example, we may take column number 2900. Okay, we're going to take that column, and that's going to be our first sample of this alignment. Okay, so the second column, we're going to pick another random column to sample. So we may pick column number 1500, and that's going to be our second column in our bootstrap alignment. So we're going to continuously sample these columns over and over and over again until we get an alignment the same length as the original. So if the original alignment has 3,000 sites, the first bootstrap replicate will also be 3,000 sites, but we're just going to sample each column with replacement. So we can sample the same column multiple times whenever we do a bootstrap. And then we're going to build a tree given that iteration. And then we're going to do it again. So each time we build a bootstrap replicate, we're resampling the data with replacement. We're resampling these columns with replacement until we get an alignment the length of the original and rebuild the tree. And so we're going to do that like 100 times. We're effectively going to have 100 different alignments constructed based off of the resampling of the original alignment. Hard to explain without drawing it out. But nonetheless, it's the most common test of a phylogeny, particularly a maximum likelihood phylogeny or neighbor joining. So I'm going to do neighbor joining because it's it's considerably faster. And so I can run a bootstrap by selecting test of phylogeny. So test of phylogeny, I'm going to pick a bootstrap. And so my default is 100 replicates. I don't want 100 replicates because I'll get the same idea with 100. So basically, I'm going to construct 100 additional artificial alignments based off of the resampling of the original alignment. And just click go. All right, and so now my tree is going to have numbers on it. All right, let me get my root out. Okay, there's my root. And so at each of these nodes, now I have numbers here. And so this at least tests the robustness of the data itself. Right? That's the best thing a Bootstrap does is it tests the robustness of the data. And so what these numbers mean, so if we're looking at the ancestral node between mammals, platypus and the other mammals, we see this value of 100. Well, what that means, out of our 100 Bootstrap replicates, we returned this relationship 100 out of 100 times, or 100% of the time. But out of those 100 replicates, human and opossum came out sister only 94 times. If we look at spotted gar and zebrafish, I get those two mixed up. Out of those 100 replicates, these two came out sister. These two nodes came out, or these two branches came out sister 100% of the time. So these Bootstrap values, they're effectively percentages of how often we saw this relationship among our Bootstrap tree, or Bootstrap trees. If we look at these deeper nodes, they become a little less robust. So it gets a little bit harder to accurately reconstruct older relationships, right? So like 55 times out of 100, we got a relationship we got a sister relationship between birds and mammals, or the sauropsids and the mammals, right? 64% of the time, green anole and zebrafinch came out sister. That's not a good value. So Bootstrap values, we only like them if they're really above 90. We're really happy when they're above 90%, because that suggests that the data keeps, no matter how we resample it, we keep getting the same relationships out of it. It's a very strong data set. It's a very nice tool. And so we can bootstrap both the neighbor joining and maximum likelihood, and we can bootstrap them for as many times as we want. It's not unheard of. It's actually pretty common to see bootstraps get into the thousands. So people will run 1,000 bootstrap replicates. Like back in the 2000s, the early 2000s was 100, just because it took forever to run 100. But now the software is so fast, the computers have gotten so fast that running 1,000 bootstraps takes almost the same amount of time, so might as well just run 1,000. All right. And again, we can save this Newick. Yeah, export tree as a Newick. Save my branch links, save my bootstraps, because those are important. Because whenever I publish, if I want to publish this tree, I would have to have the bootstraps on it. If I just present a tree and there's no bootstraps on it, nobody's going to believe that tree. Like because there's no test of the robustness of the data to make this tree. And then I would just save that as some file. So save as, and again, I want to name this as accurately as I can. So I might say Peewee BS 100.Newick nj for neighbor joining, because now I know this is a neighbor joining, and I bootstrapped this 100 times in WK. And so this NUIC, this might take 2 seconds. Let's see if I can get this done in 2 seconds. So what a NUIC kind of looks like is if we have, it's going to use parentheses and commas. So these two things are equivalent to each other. Okay, and so that's what a NUIC represents. It's a text representation of a tree where we parenthesize things that are closely related to each other and separate commas for individual species. So that's what a NUIC represents. It reflects the tree, but in a text format that computer programs can read. All right. And so let's see, we're at 41 minutes. So Monday's President's Day, but I'm not going to, so we don't have any classes on President's Day. So I'm not going to stream on Wednesday. So we'll be back on Wednesday. So no class Monday, but we'll be back on Wednesday. And I may throw up a video where I can draw stuff out, some things that I actually wanted to draw out without streaming it. I may just record it and upload it, so we have it. At least we have it. Otherwise, I'm just going to stop there, and we'll pick up. So we're going to start Linuxing on Wednesday. So I'll send out some software to help us connect to our server. Because we're going to basically do the same thing that we can in Mega on a server. And again, there's advantages to doing on a server. Workflows can be easier. Our software can be more sophisticated. So we'll start playing around on Wednesday. Until then, email me if you have any questions.